Hi, my name is Ashton Wright, and I will be teaching you about gel electrophoresis and its application to DNA analysis and forensics. Gel electrophoresis is a method used in a laboratory setting to separate DNA, RNA, and proteins according to their molecular size. The DNA separation is based on the size of the fragments and depending on their molecular weight, the rate of migration. DNA consists of a nucleus that houses nucleotides. Nucleotides consist of phosphates, which are negatively charged. As you can see here, in the pink is a nucleotide, and in the green are phosphates. The phosphates that are part of nucleotides are also part of the sugar phosphate backbone of DNA. Gel electrophoresis allows for the migration of the colloidal particles through an electrical charge to travel through a gel. This gel is called agarose. Agarose consists of a polysaccharide polymer, or also known as carbohydrates. One end of the machine includes holes which are named wells. Negatively charged DNA is placed into the wells. Since positives and negatives attract, the negatively charged DNA, or anode, will travel to the cathode or positive side. To analyze a DNA sample, a restriction enzyme is used. This is what cuts the DNA up into pieces in specific places. This can be used when determining a relationship between a parent and a child. So this is a very simple diagram of a gel electrophoresis machine. This is the wells, and this is the negative side and the positive side. This is a more complex example, and these are the fragments of DNA. And the shortest fragments travel farther to the positive side because of their weight. And the agarose is also combined with the buffer fluid. To determine the veracity of evidence in an investigation, DNA analysis is used. It makes DNA fingerprints of the crime scene and suspects. For example, in a homicide case, this method could be used to determine if a suspect is innocent or guilty. Usually, an analyzer will be given 3 or more samples of DNA. One of the samples will be the suspect's DNA, and the others will be samples of DNA from the crime scene. Then, using a restriction enzyme, the analyzer can evaluate if the identification points are similar to the suspects. The results can then be used in trials. In this example shown, suspect 3 would be proven to be at the crime scene. You can see this by how the crime scene evidence matches suspect 3's DNA perfectly. As you can see, the other suspects do not match whatsoever. None of their lines are equal to each other. So suspect 3 would be known to be at the crime scene. This is my works cited page, and I thank you for watching, and I really hope you accept me for this scholarship. Thanks.